Hello, I'm going to go over part B, chapter 11, the rest of the uh, action potential that we discussed earlier. Couple of things in mind is that uh, before we go on and talk about the important concepts, uh, you really need to review the activity of leak channel we discussed last time. If you recall, we mentioned they were located in the axon, and two of the most important ones were sodium leak channel and potassium leak channel. They're always open. More potassium was leaking out, if you remember, and primarily they were located on the axon. We also need to review voltage sensitive gates. Uh, voltage gated channel, all right. Again, we discussed several of them, sodium, potassium, and also calcium. Those were the three important ones. And we mentioned that sodium was in the axon hillock, and they causing all of a sudden for the transmission and initiation of the action potential. And then voltage gated potassium channel was a slowing opening and a slowing closing. And then calcium, primarily voltage gated channel, were on the axon terminal. So in conclusion, we decided to think that, they are located on the axon hillock, they're located on the axon itself, and they're located on the axon terminal respectively. Okay, so these were in the previous lecture if you wanted to think about it. We also talk about the role of sodium potassium pump. We mentioned these are located everywhere in the cell body, in the dendrite, in the axon, in the axonal terminal, and again, it pumps 3 sodium out and 2 potassium in. This was huge and important in maintaining the concentration gradient and also restoring the balance. Remember, they do not operate via the neurotransmitters or anything like that. They only operate via the ATP from the mitochondria. Then we discussed about the chemical gated channel. Chemical gated channel, also known as ligand gated channel. Ligand gated channel or chemical gated channel, you have some familiarity because we discussed about the neuromuscular junction, if you recall. And also a chemical gated channel can exist in the nerve when they communicate with one another, all right? So mostly these chemical gated channel on the nerve, you're gonna see it on the dendrites, as well as on the cell body, or also known as soma. Okay, so in reality, what we wanted to discuss is mostly right over here. So to give you a better perception, you have 2 neurons in communication at a time. Now, don't get me wrong, if you think of it three-dimensionally, there could be a neuron above this neuron, there could be a neuron below, there could be a neuron coming in from here, there could be a neuron coming in from everywhere, basically. But to break it down and find out physiologically what happens and learn about the mechanism of action, we're just going to look at 2 neuron in communication to make it simple. And these are the nerve. And again, I assume you guys can understand that I don't want to spend too much time and draw this in detail, but generally speaking, the first neuron that triggers the action potential, all right, this usually referred to as presynaptic neuron. So this would be pre means before, synaptic neuron. And the next neuron that receives this communication happen to be postsynaptic. What happens if you have three, or what happens if you have one above that? Well, again, this could become a presynaptic, and then the next one is postsynaptic. So this one could be a postsynaptic. If so, then this one would become a presynaptic. So it really depends, but let's not confuse ourselves, and let's talk about just two neurons at a time, all right? And find out the important concepts. All right, so this area right over here is known as synaptic cleft. Synaptic cleft is the area in which the communication takes place. The communication itself, so this is the area, refer to this area, all right? The communication itself, in other words, one communication of the presynaptic neuron with the postsynaptic neuron, all right? When these two are communicating with one another, this is called, it's making synapses. All right, that means they are communicating with one another, all right? So remember, in here, there were a lot of neurotransmitters, and that's what we left off talking about, the neurotransmitters. There were different kinds, and today we're going to explore a little bit more. But these neurotransmitters communicate with the nearby neuron, all right, or postsynaptic neuron. In general, there are two types of synapses. One is called the electrical synapses, and another one is called the chemical synapses. All right, so we're going to be concentrating on the chemical synapses. I'm just brushing off the concepts of the electrical synapses to just kind of like giving you an idea of what's going on. Electrical synapses, they don't have this delay. They don't have this space in between them. So in a way, you have an axonal terminal of one neuron that is communicating with another neuron. Maybe I should put this in a different color so you could see it better. So here, maybe a soma or a dendrite of another neuron, and you can see that they come into a close contact of one another. So if we wanted to kind of like take a look at this portion, the neuron, all right, and we could go ahead and take a look at the axonal terminal of another neuron, all right? So here is the axonal terminal of another neuron right here. So I just expand this a little bit further. There are channels. These are kind of like a gap junction, if you remember. If not, review your concepts of what was a gap junction. So basically, as the sodium going in, we mentioned, and potassium is leaving, and this sodium going in causes the depolarization earlier. And these cations, they can actually go to the nearby. And again, if there are enough cations that are passing by, it can trigger the action potential, and the action potential can fire. So there is no really much of a delay in here because they're kind of like a stuck. And there are transported proteins that are linking the presynaptic with the postsynaptic, all right? So the process of action potential over here is the same. In other words, you had sodium voltage gated channels predominantly in the axon hillock that certain thresholds, -55 is met, and then boom, they're open, and then again, action potential can trigger, etc. But that's not really what we're going to be talking about because we wanted to go over the chemical synapses. So if we go ahead and take a look at the ligand gated channel or chemical synapses, and we could pick that up from there. In other words, here you have the axonal terminal, all right? And axonal terminal, if you recall, they were, you know, and again, they could be a different type of neurotransmitter. In the muscle, we just looked at one, but in reality, they could be different type of neurotransmitters. In here, in muscles, there were only acetylcholine. Now, the idea of how cells can actually make these neurotransmitter, these are vesicles, and inside of them contain neurotransmitter, which again we abbreviated as NT. NT is being the neurotransmitter. And if you recall, the action potential was coming, all right? As action potential was coming, all right? It opens up the calcium voltage gated channel. Pretend these are the calcium voltage gated channel. Calcium would enter, all right? And then again, that caused the exocytosis with the use of ATP, all right? You could release this neurotransmitter outside in the synaptic cleft, and now you have a bunch of neurotransmitters on the synaptic cleft. All right, notice that in this case, I'm putting two different kinds. But more importantly, how are these neurotransmitters are actually generated by the cell? The idea is simple. If you remember, every cell has the DNA. So DNA can make the messenger RNA, if you remember, and the messenger RNA can be involved in making proteins. And these proteins, they can go to the Golgi apparatus, and the Golgi apparatus can actually direct them into different areas. And that's what they are. These are different type of neurotransmitter, but many of them are protein-based, and that's how they are being built, all right? So when the cell is releasing them, you might ask, okay, well, now they don't have any more neurotransmitter. How can they send the signal again, all right? Or they become useless? No, because the cell can generate more neurotransmitters. And also, if you recall, they can actually reuptake some of these neurotransmitter back so they could reuse them. Sometimes they are broken and they need to put them together, but there is a recycling going on. And this phenomena we mentioned was reuptake mechanism. Now, sometimes you need a special channel protein to reuptake them. Acetylcholine, for example, was one. When you break down the choline and acetic acid, choline molecule was reuptaken back into the nerve, so that way you could recycle, and we mentioned that this becomes more efficient, if you recall. All right, so what is happening in general is action potential is firing, all right? And as a result of that, this causes the release of neurotransmitters, which are right here. This neurotransmitter is now going to have to bind to the receptor, okay? Neurotransmitter bind to receptor. So again, there could be a specific ligand-gated channel, all right? So let's pretend that this is a post-synaptic neuron. And again, I'm just gonna use, although they could be in the dendrites as well. Let's say this is the dendrites, and these are the cell body, all right? But and here's the axon hillock, and here's the nucleus. So the channel proteins, ligand-gated channel could be here, all right? Could be, you know, on the soma. All right, let's put in a few of them. The one that I'm showing in red, it just means that, you know, the receptor is this, oh, I should have put it in blue. All right, so the one that I'm putting it in blue, and then let's make this blue, all right? And so on, so that the neurotransmitter can bind in here, all right? And the black one, again, is a different neurotransmitter. So these are unique for a different one. So these receptors are specific, all right? So after it binds to the receptor, right? Then this could cause either triggering action potential or inhibiting action potential. If the action potential is triggered, obviously the communication is going to go on, and you know, and the action potential is going to fire, but not necessarily all the time that is going to happen. All right. So remember, because there is a gap in here, in the synaptic cleft, there is a space in here, and it takes time for this neurotransmitter to bind the receptor and then open up this channel protein and then allow certain ions to go back and forth. This, we call it a synaptic delay. All right. So this part right over here refer to as synaptic delay. Remember that neurotransmitter needs to bind to the receptor. So first they need to find their specific receptors. Then as soon as the receptor, the neurotransmitter was bind to the receptor, now they can open. Now ions can go in and out. So that all will take time, all right? That will take some time. Okay, let's go ahead and take a look at a few other concepts, and then we come back to this part. Just wanted to let you know that the way in which a communication can take place is so many different ways. Like a lot of times we mention axosomatic. What does that mean, axosomatic? That means axon. Here is the axon of one neuron. This is the axon. Communicates with the soma. Soma means cell body of another one. So this kind of communication right here is an axosomatic. Axon of pre and the somatic of post synaptic neuron. All right. So there could be axodendritic. All right. So if these are the dendrites, for example, all right, and you have, another neuron with its axon, for example, coming in and communicating in here. Let's say this is a neuron and this one is axon. This would be axodendritic. Axodendritic means axon of one neuron, presynaptic, communicate with the dendrites of another. So you might find actually so many different ones: axonic, axodendritic, axosomatic, axonic. All right. So again, it just tells you that the communication can take place in many different ways. Again, some of those could be electrical, some of them are chemicals. All the chemical ones happens via the axosomatic and axodendritic, and that is what we wanted to focus on. All right, so keep that in mind. All right, next up, the electrical synapses are less common. And again, they go through the gap junction. This is important in central nervous system, like mental attention, memory, homeostasis of water regulation. So maybe if we get the time, we can discuss this later. I'm not sure, but again, that's a little bit easier to understand because again, the axon, it just the action potential goes from one to another. This chemical synapses is a little bit more complicated, and again, you had a little bit of this idea before, so it makes more sense to talk about that in a more detailed way. All right, to understand what is the overall picture. The overall picture happened to be very simple. I mean, you're trying to communicate. You're trying to communicate because if you recall, we mentioned you have the sensory receptor, then you have control center. This mostly in the brain, and then you have the effector. So in order to respond to a particular stimulation and respond and send a signal to your organs, information needs to be received first, and information needs to go certain places. So you can see how the neurons can now communicate. If you perceive something as a touch, like you're touching a particular surface, let's say it's a hot surface, the nerve has to communicate now. All right, so it's going to send a signal to the control center. From there, motor neuron is going to send its signal. Again, how the signal is being sent is right here. It's right here. One neuron communicate with another. If you remember, we call this sensory neuron. Sensory neuron sense things, send a signal to the control center. Motor neuron. This was the neuromuscular junction that we discussed. All right, and in between, we had the interneuron. There could be many interneurons communicating, pretend these are the neurons inside of the brain, for instance, all right, and transferring information from one area to another, all right? So that's what we wanted to get at literally, all right? But neurotransmitters and the receptors mediates the change in the potential membrane, and it's really what that means is depends on the type of neurotransmitter and what they are releasing and what of these channels are opening and how much neurotransmitters we have. There are many channels. I just put in 4 channels in here, all right? So the amount of neurotransmitter bounding to the receptor is one of the key factors. If you have a lot of these, let's say, darker neurotransmitter and they're all binding to the receptor and they are opening them, that makes a big difference about which ions are going to go in more or less, all right? So, so how many of them are available is another thing. How many receptors do you have? Again, receptors are proteins, so this cell can make them via the DNA. So a lot of things plays an important role to find out the effect of that particular neurotransmitter. But 2 things so far that you wanted to remember is the number of neurotransmitters is important. Obviously, if you have less, they have less chance of binding to the receptor. Number of receptors are important. How many of them do you have to begin with, all right? And how long does it take, the amount of time that the neurotransmitter is binded to the receptor? If they're binding for a longer time, this channel is going to remain open. If the enzyme comes in and break it down right away, the channel is going to be closed, all right? So again, how long, how much enzymes do you have? These are also important. So how long neurotransmitter, all right, is bound to receptors, all right? So that is another important concept that dictates the changes in the membrane potential. In a nutshell, there are 2 kinds of postsynaptic potential. In other words, you have one nerve, all right, and you have another nerve, and this is postsynaptic neuron, if you recall, all right, this one right here. Post means after, synaptic. They're making synapses right here, okay, potential. So there's 2 kinds. One is called EPSP. EPSP stands for excitatory postsynaptic potential. And then you have IPSP. IPSP stands for inhibitory postsynaptic potential. So basically, what you wanted to get at this is the EPSP, they use the chemically gated channel, all right? And action potential is more likely to occur. Excitatory, that means usually what happens is they can initiate the action potential. So action potential is more likely to happen. And the opposite, inhibitory action potential, is less likely to fire. Okay, so we're going to go ahead and take a look at excitatory postsynaptic potential. Again, if you put your voltage meter and take a look at the a signal that, let's say, is excitatory, maybe we can kind of like look at this in a greater detail. Huh? Okay, so let's go ahead and take a look. All right. So here is the excitatory postsynaptic potential. All right. And this arrow, red arrow, indicating that the signal has been sent. Obviously, you can see it didn't reach the negative 55, so this signal, did not cause the action potential, but it went really close. So if you're right here now, let's say the neurotransmitter binded to its receptor opens up, and certain calcium or sodium going inside. Sodium is positive ion, but very soon enzyme is going to break it down and that channel is going to close. Not enough sodium went in to cause the postsynaptic neuron to get to the negative 55, which was the threshold. But it was an excitatory signal because of the fact that action potential is more likely to happen. If it happens, it's still excitatory. If it doesn't happen, but it was about to happen, or anything, going towards the positive direction, it would be excitatory, all right? Because action potential is more likely to happen, all right? So let's go ahead and take a look at a scenario in which this could happen, all right? So let me see if we can find the appropriate, or maybe I just should draw this. Okay, so we're looking at the excitatory right now, okay? So if you have the action potential is coming in, and let's say neurotransmitters are releasing, and this neurotransmitter, let's say, is excitatory neurotransmitter. And as a result of that, they're binding to the receptor. And as they're binding to the receptor, is opening up a channel. And this channel happened to be for the sodium. So now sodium is moving inside. All right. Now this happened to be the postsynaptic neuron. All right. In our previous example, it still inside became a little bit positive because you had negative 70 and millivolts in here. That was the resting potential. But now is, let's say, negative 60. All right. That's not enough to cause the action potential, but it's still, because it got positive and action potential is more likely to happen. All right, we call this excitatory signal. Now, chances are this could be broken down by the enzyme, and as soon as that happens, this channel closes. So let's close this channel now. No more sodium is going to come in. All right, now it is completely closed. All right, so now the channel is closed and no more sodium can go in. And as a result of that, what will happen in here? Well, in here, it was now, I don't know, negative 60 millivolts. But pretty soon, again, the sodium-potassium pump working and kicking 3 sodium out and bringing 2 potassium in through their channels and so on. And guess what? This positive channel is going to get restored back to the negative 70. So that's exactly what happened in here that I showed you. All right. So besides, there were some leak channels as well, but notice that again, resting membrane potential achieved. All right. So these are all excitatory signal. Going back to the previous ideas, all right. If you recall, how can we get rid of these chemicals in here? So there were 2 mechanisms you're familiar with. One was degraded by enzyme. A specific enzyme in neuromuscular junction was acetylcholine esterase, if you remember, all right? But it doesn't have to be. There could be different enzymes. Enzymes can break this. So that's one thing. The more enzyme you have, the faster you can break these down and the channel is going to close. Lesser enzyme, they might remain open. All right, so it really depends. Again, the number of neurotransmitter, how many of these are in here, how many receptors you have, all of those would dictate and mediate the effect. Either way, one way to get rid of this is via the enzyme. Another way, we mentioned that sometimes it's not like a magnet that they can attach and not disattach, so they could just depart. So they could also depart. The neurotransmitter can depart away from its receptor, all right? Once they move away from the receptor, same thing is gonna happen, all right? Another thing, they can be reabsorbed by the presynaptic neuron. And again, I think if I recall correctly, we mentioned about the reuptake mechanism. So sometimes as the enzyme break it down, the product, sometimes this neurotransmitter, so it can get reuptaken back. So in other words, you're recycling things, you're putting the neurotransmitter back into these vesicles, and then again, ready to for action potential to come in order to be released and so on. So another one is reabsorb. This is sometimes is known by reuptake mechanism. There were a lot of supporting cells as well. So some of those supporting cells, if you recall, were the astrocytes. Astrocytes, they look kind of like a star shape, all right? So again, some of these star shape looking and so on, they can also absorb that as well, okay? So few mechanism, enzymes, just normal departure, reabsorbing or reuptake mechanism, and finally the astrocytes. Those are all different mechanisms to deal with the neurotransmitter that was in the synaptic cleft and available. All right. So now I'm going to go ahead and give you an example. All right, of the inhibitory postsynaptic potential. By the way, one of the good example of the excitatory postsynaptic potential is actually acetylcholine in neuromuscular junction. But you have to be careful with that because you know, it really depends. Acetylcholine was excitatory in neuromuscular junction. ACH in neuromuscular junction was excitatory. So that was a good example of excitatory postsynaptic potential. All right. Glutamate is another one. You will find out that the glutamate plays an important role in like your senses, such as olfactory, for example. Right? They communicate one nerve with another via this glutamate. All right, so these are excitatory. They can cause depolarization. Not necessarily action potential, but they can cause depolarization. All right, now let's go ahead and take a look at inhibitory neurotransmitter. Inhibitory neurotransmitter, IPSP. All right, two good example of that is GABA neurotransmitter and glycine. All right, so they cause, instead of depolarization, can you guess what it causes in terms of your resting potential? What do you think these type of neurotransmitter, these are neurotransmitter? Glycine and GABA. And IPSP, it's inhibitory. Action potential is less likely to happen. So my question to you is, can you imagine, or can you see if you can understand what is these two neurotransmitters could cause? All right. You could pause the video and think about it if you want. All right. But the resting membrane potential, it causes to become what? Okay. Hyperpolarization. If you got that, congratulations. All right, you got it. In other words, action potential is going to be less likely to happen. So we go ahead and take a look at this phenomenon right over here. You could see the stimulus is now hyperpolarizing. In other words, opposite of depolarization. You're going towards maybe, I don't know, -80, -85, somewhere around there. So this signal, whatever that signal was, caused the membrane potential to become more negative. All right. So if you have the presynaptic neuron communicating and releasing their neurotransmitter, possibly GABA, okay, and this postsynaptic neuron have their channel now open because the neurotransmitter binding here, okay, and now this channel is open, all right, and this cause this to be negative inside, all right, that's what it's saying, all right, so obviously it's not for sodium ions to go in. If the sodium ion went in just like the EPSPs, then it would have been positive. But in this case, it's negative, right over here. All right. So I'm going to be elaborating a little bit to see, and then I give you an example of why do we need inhibitory and excitatory postsynaptic potential, or inhibitory one, to begin with. What's the big deal about it? All right. So let's go ahead and take a look and draw this separately so you could see it a little bit better. All right, I'm going to be using a different shape of neurotransmitters just for the sake of it. So if you have, for example, exocytosis, and you have this neurotransmitter that looks triangular shape, let's just say, all right, and is binding to the postsynaptic neuron. And as you bind it, I'm just going to leave this channel open, all right, because I assume that they already binded in here, right? These are ligand gated channel, all right? So, and this is part of the, postsynaptic neuron, all right? Now I wanted to demonstrate what is happening in this case, all right? So let's say that these are for GABA, all right? Let's put in another neurotransmitter also, all right? So this is example of IPSP. But what if we have another neurotransmitter and so on? Let's say this one is glutamate. We'll look at this later. But right now, I'm going to focus on the GABA because I already talked and give you an example. All right. So let's say GABA is binding to the neurotransmitter. All right. And inside was negative 70 millivolts. That was the resting potential. And pretend that you have a voltage meter and you're looking at this resting membrane potential. But as GABA comes in here, it could be a chloride ion now. Chloride ion, this is for chloride ion. Because they are anions, they cannot get into the membrane, right? They're not permeable. But what if chloride ions now can go in, all right? Because their channels are open now. If the channels are open, chloride is negative. So this becomes more negative now, all right? So maybe it becomes negative 85 millivolts now. Pretty soon, neurotransmitters, they're going to, enzymes is going to break down these neurotransmitters as this channel is going to close. So they're not going to, hyperpolarize forever. There are also some leak channels that they could balance itself. All right, so there are many different mechanisms. Now, simultaneously, let's say that another neurotransmitter, glutamate in this case, are also opening up their channels. All right, so here it goes, and the glutamate binds in here. Now, let's say this is for sodium. Now, sodium is going in and causing the depolarization. Wait a minute, so this one caused hyperpolarization, this one depolarization. In this case, it looks like it's more chlorine is coming in because I'm putting more channels in here, so there would be more negative compared to the positive. But if you have many of these, then it could be positive. So basically, in the soma right here was a soma, all these charges, and because of the different ions coming in and out, we can calculate in here and figure out what would be the outcome. Is the outcome is going to be inhibitory signal, or is the outcome is going to be excitatory signal? But before we do that, let's go ahead and give you guys a problem and see if you can solve this, and then we'll come into the these two signals again, and we'll talk about the process of summation. All right. So if you're adding different numbers, you're saying like, you're summing. Summing, that means what? That means you're adding things. So there is a process of summation. I'm going to get into the summation a little bit shortly, okay? But right now, I wanted to give you guys a problem so you can guess, or you can see if you can solve this problem to test your understanding, all right? So let's say unknown neurotransmitter, don't know what shape should I use now, but how about asterisk, all right? Unknown neurotransmitter, or a star shape, all right, is being released from presynaptic neuron, all right, and is binding to the receptor of the postsynaptic neuron. So pretend that these are a star shape, for example, and they're able to bind these, and as they bind them, their channel is opening. And these are the postsynaptic, huh? This is a neuron. Now, pay attention. All right, pay attention. Before that happens, let's analyze in here. Earlier, we mentioned that because of the sodium-potassium pump, you always pump 3 sodium out and you bring in 2 potassium in. So again, this is related to the previous chapters. If you don't know these concepts, you'll be lost. So you gotta learn the concept that you learn and the process of action potential in the first part. What does that do? What it does, increase the concentration, bracket means concentration, of potassium ion in here and lowers the concentration of sodium ions in here. And vice versa on the outside, lowers the potassium concentration and increases the sodium concentration. This was the case in the muscles as well, right? Now, this one is opening up, and this problem is saying whatever that neurotransmitter is, pretend it's a neurotransmitter that is unknown, right? This neurotransmitter is for ligand-gated channel is for potassium ion, all right? So once it opens, only potassium ions can pass through this channel. My question to you is, do you think this neurotransmitter is a IPSP signal or excitatory or inhibitory? That is this particular neurotransmitter. Remember, problem is stating is for sodium, is for potassium ion. These are potassium ligand gated channel. So what that means is when it binds, now potassium can only go through these. See if you can guess what it is. Remember, inside of here, we're starting with the negative 70 millivolts, and that was the resting membrane potential. All right, if it's for potassium. What do you think? Is it excitatory or is it inhibitory? Pause the video, see if you can guess, or see if you can make a sense of it. All right, so because outside of the nerve is lower potassium, and when it binds in here, and if it's for potassium only, guess what? Here you have a potassium. So potassium is gonna go from high to low, okay? This was facilitative diffusion that we discussed, all right? Using a transport protein, they're going in there. Now, inside of the nerve, you have less positive, because a positive ion is exited. So you had some positive ion exiting the cell. Well, obviously, if you have a positive ion exiting the cell, what do you think is going to happen to this negative 70 millivolts? It becomes more negative. So this is IPSP. Why is it IPSP? Because positive ion leaving. So no longer you have a negative 70, all right? You have more negativity inside because the positive ions are leaving inside. So you have more negative, all right? So action potentially would be more likely, less likely to happen, and that's what will happen. So anyways, if you got that, congratulations, good job. And if you didn't get it, that's okay. At least now you know what is going on and so on. Now, I have to add few things, and you know, sometimes maybe you'll look at it a little bit later on, and you'll have, for instance, a nerve underneath your skin, and if you put in a certain pressure, these channels might get open. And as it gets open, let's say sodium can go in now and cause the depolarization, and that's firing the action potential. And now you know you touch something. So sometimes pressure cause this pressure sensitive channel to open up, and then permeability for the ions can change, all right? So, you know, a lot of times the touch receptors act this way as well. That's why you can, you know, feel something is penetrating your skin, etc., because underneath your skin there are a lot of different channels, and they can open and close. Similarly, sometimes this can happen with the temperature. All right, the fluctuation of the temperature can open and close certain channels. But again, that's not really what we're going to be focusing. We are focusing on the IPSP and EPSP. I just want to throw that in, so give you guys a perception of other things can happen. Let's go to the summation. Remember, the process of summation is when things are adding up. There are two kinds of summation. You have a temporal summation, and you have a spatial summation. All right? It is important to know the difference between the two. So let me define the temporal summation. This is when presynaptic neurons transmit their impulses or action potential in a rapid fire order. Boom, 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 boom. They're sending the signal pretty quick, all right? So they can add up quickly. Versus the spatial summation, postsynaptic neuron is stimulating by a large number of axonal terminal at the same time. Let's go ahead and take a look at some of the pictures to help you understand these two mechanisms. So right over here, you have no summation, all right? What is happening? There is a excitatory signal coming in from the presynaptic neuron. This is a postsynaptic neuron, but it's not enough to cause the action potential. You could see it's probably positive 14. It's a little bit shy to reach the negative 15, which was the threshold. And then again, sodium potassium pump restore the balance. And then again, once you're reaching complete resting potential, the same signal is fired, and again, that's not enough. The reason why it's not reaching is because these two are two signals exactly the same, positive 14, are farther apart from one another. But what if one signal is coming, but soon after there is another signal is coming? So here is the same signal, but there are closer intervals with one another. Temporal, tempo means time. So again, it's a impulses in rapid fire order. What that means is the E1 is coming, releasing neurotransmitter, but before they show the complete action and before the sodium potassium pump restore the balance, another same signal is coming in. So this is adding up now. Because it's adding up, it's reaching the threshold, and as a result of that, that's enough to open up the sodium voltage gated channel that are predominant in the axon hillock opens up, and therefore the action potential is firing in this case. All right, so this is called a temporal summation. Time is important. A spatial summation is when you have one postsynaptic neuron is stimulated by large. Like here, you have one signal, here's another signal. They come in at the same time. So if they're coming in at the same time, let's say this one has positive 14 and this one has positive 14, together they come in, all right? And there's like, let's say one of them is opening up a lot of sodium channels. Sodium goes in and becomes depolarized, all right? But not enough to cause the action potential by itself, but another one is coming in, opening up more sodium channels and so on. So now is enough, these two adding up to reach the threshold, and again, action potential fire. Remember, the action potential, whether it's firing or not, that's not really the concept here. The concept is, in this case, these signals are adding up because they're coming in at the same time. Well, in this case, for example, what if this signal was a positive 14 and this one was a negative 14 and they came in at the same time? Then they neutralize one another. They neutralize one another, and in this case, action potential did not occur. No action potential in this case, because it never reached the the threshold. So, but it still was a spatial summation. All right. So a spatial summation, in this case, or let's say when the many signals coming at the same time, you know, it doesn't mean whether the action potential has to fire. Maybe, maybe not. Let's do a problem for you. I wanted you guys to see if you can do the following problem. Here you have a postsynaptic potential. All right. Instead of neuron bringing their signal and neurotransmitter, which can happen, we can put in the electrode in here and zap this neuron. All right. What if we zap it with the following at the same time? At the same time, they're all linked to a different voltage generator. This one was a negative 10 millivolts. This one positive 5 millivolts. This one negative 7 millivolts. This one positive 15 millivolts. This one, let's say, is positive 7 millivolts. All right, because they're all come at the same time, what kind of a summation do you think this is an example of? You're zapping this neuron all at the same time. Could pause the video and see if you could guess. Your second question is action potential going to fire? All right, so in order to figure out action potential is fire or not, we gotta add these numbers together right here, and that would be in the soma. But the phenomena is a spatial summation because one postsynaptic neuron is being stimulated all at the same time. So it looks like positive 7 and a negative 7 cancel one another, so we could just cross this. Right here is a positive 15. This is positive 5. That's positive 20. And then in here, you have a negative 10. So if you have a negative 20 and positive 20 and negative 10, that gives us the total of positive 10. What are we starting at? We're starting at negative 70 millivolts. That was the resting potential. Now you have positive 15. Which will take us to negative 60 millivolts. Is negative 60 millivolts enough for action potential to fire? In this case, no. Action potential does not occur. All right, it gets close. All right, so yes, overall, it's a excitatory stimulation, but is not enough to cause anything really. All right, so that's the concept of the summation. Now, the next concept I wanted to tell you guys that certain neurotransmitter, they may have two effects. So far we looked at certain neurotransmitter to be inhibitory and some neurotransmitter to be excitatory, all right? But some neurotransmitter could have both, depends on where we're talking about. Now again, neurotransmitters have different receptors. Acetylcholine, ACH, is actually a good example of that. Inhibitory, we discussed, that would be a glycine and GABA. Excitatory, we discussed some of them already, like glutamate, all right? But acetylcholine could be both, all right? In fact, is excitatory in neuromuscular junction, but it's inhibitory in the cardiac muscle cells, all right? So we could slow down the heart instead of accelerating it. And we'll discuss that a little bit later on in chapter 14. So it could have both, depends on the location, and more importantly, because there are different type of receptors and therefore they could do different things, all right? So that was another concept. All right, there have been many neurotransmitters have been identified, serotonin, norepinephrine, epinephrine, you know, so there's so many different kinds and you could look those up. Imbalance of these neurotransmitters could cause a lot of different problems. including psychological problems, including temper, including lots of addiction problems, etc. So again, it would be very interesting to look at this when you have the time and take a look at, for instance, endorphins and what they can do, and people that are addicted, for example, what happens that are addicted to cocaine, for instance, and such. All right. But at this point in time, we're not going to go into those disorders. All right. Also, that would be a great topic that you wanted to take a look and write and read about. All right. So next up, what we wanted to do is take a look at G-link protein.